Now, the experiments that I've described actually take thousands of mice. So this year, I bet we will have collected about 20,000 animals. I run my lab, I think, and I read a lot of business magazines, much in the same way that Dell makes computers or Toyota makes cars. We obsess about an efficient pipeline. I obsess about cost per mouse. And so what we can do in this lab is basically experimental epidemiology. If you come to my mouse colony, you will notice that every single mouse that's born lives under the same conditions. So the mothers eat the same food, they live in the same cages, they get the same amount of sleep. They don't get infections because they have a clean colony, so viruses, which are a cause of human heart defects, are not a factor here. The only three factors that vary between the animals, as far as we could tell, are the age of the mother, the age of the father, and the size of the litter that the baby with a heart defect is born to. Now, and so we can do a statistical analysis to figure out which of those three variables might or might not be important. When we discover, in this particular case, well, actually in all cases of all different types of heart defects, is that the age of the mother is really important. This has been shown in multiple epidemiologic studies, but not well understood why that is. The most common explanation, or if you ask a medical student or any doctor why this is, they will tell you that it's something like Down syndrome. So what Down syndrome is where a mother gives two copies of the same chromosome, chromosome 21, to the baby, and this relates to the age of the oocyte. The oocytes aren't able to develop normally. Well, in a number of experiments, we eliminated that, basically what I think is a trivial explanation. So that means there's something about the age of the mother that influences how the baby's heart develops. There's a factor related to age. It could reside either in the mother, like in the womb, or it could be related to the oocyte. The reason why it can be related to the oocytes is that all the eggs that a mother produces during the childbearing years actually were formed when the mother was an embryo. So how do you tease apart this problem? We do an experiment that will never be done in humans. It could never have been figured out by epidemiologists. What we do are ovarian transplant experiments. So I can take the ovaries, the eggs, from an old mother and put it in a young mother, and vice versa, from a young mother and put it in an old mother. And from here, we can collect all the babies born from these mothers and figure out whether the incidence of heart defects tracks with the age of the mother or the age of the ovary. This leads to Plan C, the bonus Plan C. So we've done this experiment now. We've collected probably nearly 200 or more than 200 babies, and the statistics are quite clear. Here in the open bars is the incidence of heart defects that we see in babies born to old mothers with young ovaries, so it's about 17%. And in young mothers, it's about 5%. And we can do math, assuming that the factor resides either in the oocyte or the mother, and predict what the result would be. So if the factor was related to old eggs, then you would expect the incidence to be quite low in the old mothers and the opposite in young mothers. If the factor resides in the mother, you would expect a high incidence. And the results are really black and white. The incidence of heart disease that we observe tracks with the age of the mother. So we have eggs being born here that are way beyond natural. There are eggs that are 600-700 days old, right? So you never see a 700-day-old mouse produce a baby. And so it's quite clear that there's something in the mother that affects cardiac development. Now, I can't fix the birth date of a mother, right? That's fixed in time. But there are probably factors that we can address. So what changes as you get older? We've noticed that mice get fatter as they get older. In humans and in mice, the incidence of diabetes, and one type of diabetes is actually related to age. Diabetes during pregnancy is related to age, and diabetes affects cardiac development. Hormones might change as a person ages. These are things that we actually understand really very well and have drugs for. And so the lab has designed a series of experiments to tease apart the relevant pathophysiology and pathways that are involved. This is plan C, treating the environmental factors. The reason why this is important is, there are many different causes of congenital heart disease, probably none of which I or any other cardiologist will ever be able to fix. On the other hand, there are these other factors, whether genetic or environmental, that have an incredible impact upon the risk of congenital heart disease. So what I show here is a plot where we just stratify the risk of heart disease. So on the horizontal axis, I show mice that either have all good genes, so no bad genes, or a bad gene on either chromosome 6, 8, or 10, or some combination of the chromosomes. And I plot the incidence of heart disease as a function of genetics and the age of the mother. So in the white bar is 100-day-old mothers, 300-day-old mothers, 400-day-old mothers. What you can imagine then is when a baby, a child, a future baby, right? So I want to change how cardiology is done. I don't want to cut a baby's chest open or stick catheters in the heart. I want to have the baby come out with a totally normal heart. What you can do is try to figure out when a woman wants to become pregnant or when a mother presents, what's important, and then logically design strategies to keep that baby's heart on a normal track. So you can imagine if a mother is very old, we might address the factor that's related to maternal aging. On the other hand, if a mother is relatively young, maybe genes are more important. Maybe I would give a medicine that affects blood pressure so that the heart stays on a normal track. Okay? I think thinking about congenital heart disease this way explains a lot more of the variability that we see and gives us more options for preventing disease in the first place. So the conclusions, the basic scientific conclusions of the work that we've done so far is that there are many other factors in addition to the main cause that contribute to the risk of congenital heart disease. So all the mice I study, they all have the same cause. They all have that genetic mutation. But you can see that the risk varies from a small number, 2%, all the way up to 20%. And these factors are innumerable. We can define them in a mouse model in a way that you could never do in humans, because humans are just too heterogeneous. You can't make a whole population eat the same food and get the same amount of sleep.
Any questions? Anybody have any questions? Go ahead.
Maybe I can start. Can you describe very briefly to folks who really don't know, what does it take to go through and define what kind of congenital heart disease you have in a mouse heart? How big is a mouse heart? What do you have to do that?
So I have samples here of mouse hearts. These are actually bigger than the baby mouse hearts. A baby mouse heart, so I collect them. Obviously babies die of heart defects, so you have to collect them really young. We collect them at about a few hours of age. It's about the size, half the size of a pea. And there are examples here on microscope slides. And what we do is we section the mouse heart from front to back, kind of like doing a CT or MRI scan of the body. And I've trained students in the lab, and a number of important collaborators who have kept my thinking straight. Thank you.